Well, good afternoon, everybody, and thank you very much for coming to the event today. This is, as Joe mentioned, this is the fourth event, the fourth symposium that we've been doing in India. We've had a great time and enjoyed the hospitality of India. So thank everybody for that. For me, the biggest one of the thing that's really pleased me is the engagement that we've had in Delhi, in Mumbai, in Hyderabad. I expect the same today from the audience, that the quality of molecular diagnostic tests is absolutely critical for getting the best patient treatment into the clinics. And it's an honour that you've been able to make the time today to come to this symposium and I guess pause your day jobs to learn more about what other people are doing, or the possibilities of setting up efficiency schemes within India. And it does I have to thank Dr. Simon Patton for joining us from the European Molecular Quality Network, as well as Dr. Purvish from the Molecular Oncology Society. So I might be going backwards now. Go forwards. It's been each day we've been ticking off the cities. So we have got the final tick. So after this, I can call my wife to let her know that I will be returning. The one question that I have been asking, and I think I ask it to myself as well, and it is an important one, for what you're doing in the labs each day. And Dr. Purvish made a point in a previous talk that it isn't just moving 5 micropieces into a tube or looking down a slide and looking at staining. It is dealing with patient samples that whatever you report onto that patient sample will lead to them potentially living longer. You could be saving their lives and actually extending their lives from weeks to months and potentially years. And the one challenge, and it's not just within India, it's a worldwide challenge that Simon will show data on, is molecular diagnostics. It probably isn't as good as it could be, and this is where driving the quality and driving the processes, driving best practices. If you can answer this question, it will set the benchmark for what you want to deliver within molecular diagnostics, and that is what level of cancer diagnostic treatment would you expect for your friends or your family members, or unfortunately one day maybe yourself. Cancer is a terrible disease, and it's very much up to all of us to do as much as we can to help patients that do have cancer. Now, Narinder was very keen that I do introduce who I am, so you have a grasp of that. This is my very short timeline. I was born on the 29th of November, which actually is the same day as Ryan Giggs. I'm not sure if I can do the same joke and get 4 times in a row, but I'm going to give it a go. That I was a very good footballer in my young days, but then tore my cruciate ligament in my left leg. So if I hadn't had that injury, you may have had somebody different here while I was training at Old Trafford for the next game against Everton this weekend. But fortunately, I did destroy my knee, and here I am today, and you've got me doing the presentation. So I then did both my Bachelor's of Science and my PhD at the University of Sussex, where I followed in the footsteps of Sir Paul Nurse, who got a Nobel Prize in cell cycle checkpoint control and DNA damage repair. From that, I moved into an antibody company, Abcam, where I was looking after the chromatin epigenetic products, and worked with Jonathan Milner, who's an entrepreneur within Cambridge. I then moved on to look after innovation within a Belgian company focused on epigenetics, and then in 2011, I joined almost 5 years ago, joined Horizon, who has got a very strong link to Jonathan Milner, who's also one of the founding members of Horizon. And within Horizon, over the last 5 years, really focused on developing molecular diagnostic controls and looking at improving the quality of molecular diagnostic assays and pipelines within laboratories. And it's been really pleasing that we've gone from working with no labs to, I think the official numbers are somewhere between 200 or 250 labs worldwide, where they're using our standards, and they've used those standards to validate assays, to improve assays, and to routinely demonstrate that their assay is working on a particular day. Now, I guess on the flip side of in this timeline, and in some ways though, I'm probably quite fortunate. I've only had 4 incidences of cancer within my family and friends. But along the journey, I've had 3 family friends who have all died of cancer. They've all had, as soon as they got diagnosed, they went through all the treatment, they got all the side effects, and really their quality of life wasn't actually that great once they had cancer. There's actually what's nice is having very recently a friend who actually had breast cancer. It developed then to brain cancer. I would say about 6 weeks ago she was given weeks to live. She was then put on Erbitux treatment. The Erbitux then actually has put the cancer into full remission, so her life expectancy has now moved from weeks into months and actually potentially years again. And this for me really is driving why I'm doing what I'm doing. There's others within Horizon that are doing that also. That we all know that cancer is this terrible disease. It touches everybody. It's not specific to specific parts of the population, and there has to be something that we can do about improving the patient treatment, so that if anyone does get cancer, whether it's yourselves, your family, or your friends, you know that you're doing as best a job as you possibly can to make sure that they have the best chance of survival and extending their life. So Joe touched on why we are here today, and these are some statistics that I've pulled out comparing the USA and India. So within the United States, for cancer deaths, there's about 50% of cancer deaths occur above the age of 50. On the flip side, within India, there's 71% of cancer deaths that actually occur in the working age of 30 to 69. And if you delve into that data even more, what's astonishing to me is 70% of people that are diagnosed with cancer will actually die in their first year from that. And that may be that because they're getting diagnosed very late. It may be that the treatments aren't available right now. There's probably, these statistics may actually be worse because there's probably unreported statistics as well. And I think for me, it's what can we do today? And I'll probably say it a few times, but I'm only one person. I've got real drive to improve cancer diagnostics as best as I can, and Horizon as well has that. But it's what can you do as well? What can you do in the lab to push that forward so that we can get earlier diagnosis to these patients and get them onto the correct treatments as fast as possible? Because that's important. By 2035, which is 20 years away, there's an expected increase of 70% of incidences of cancer. So there could be 1.7 million people within India having cancer. And if there's 1.7 million and 70% of them are all dying in the first year, that's a lot of people that we have to try and do something about. And it brings back to the question of those 70%, how many may be friends or family, and how good do you want the medical diagnostic tested when they go to the clinic, and have their samples tested. So moving sort of in a slightly different direction now. I think everyone would agree that personalised medicine is the way forward, where the perfect scenario is you have somebody with the green cancer comes in, they get the green pill. Someone with the blue cancer, they get the blue pill, and someone with the red, they get the red pill. And that's how the newspapers publish this. That if you've got this cancer, you get that cocktail. You got this. And it's good. The tricky part, as you all know, is how do you tell who needs which drugs, who needs which cocktails, and how do you develop that forward? And the key part there is you need the precision diagnostic test. And you need a test that will be able to take the population of the unknowns. We don't know what cancers they've got or what genotypes are driving those cancers, and then put it through the diagnostic labs like yourselves, and then identify whether it's a green cancer or a red cancer, and then you give them the correct treatment path. Now, what I'd like to do, and Joe did mention this at the start, is just ask a few questions to you so we can understand a little bit about what you're doing in the lab and some of the challenges you have already. Now, what we did earlier was put some coloured cards out on the seats, and what I'll do is ask a question, and then there'll be answers of your opinion, whether it's green, red, blue, green, and I'll just summarise what the results are. So, because it's important to test technology before we dive into this, I would ask if everybody could hold up the yellow card. And I have to admit that this is like the best participation I've had so far, so thank you. Somehow I've got everyone enthused. So then what I'll do is I'll ask the question, and then the answer will come up and you choose your card. So the first question is, what molecular diagnostic platform does your lab use the most? If you want to put more than one card up, you're more than welcome. It's easier for me to see if you have one card, but from experience, 2 cards is good, 2 or 3 is good. So the red card is Sanger sequencing, the blue card is qPCR, the yellow card is primary sequencing, and then the green card is next generation sequencing. So, wow, this is unusual. We've got next generation sequencing sitting on this half of the room, and we have qPCR on this half of the room. I don't know how that's, but it's good. It's interesting. It's been a mixture. It's been a mixture of with Mumbai in Delhi. Certainly, we had more people using that said they were using Sanger sequencing in Mumbai and Delhi, but it looks like there's quite a drive, or the people here are pushing into the next generation sequencing. And what's good is I've actually got some NGS data that I can show towards the end of the talk. The next question is, if you were setting up a molecular diagnostic test, which one of these is most important? Is it a molecular diagnostic test with regulatory approval in your country, which is the red card? Is it a molecular diagnostic test with regulatory approval from another country, for example, FDA cleared? Is it a test developed in your lab, so an LDT, and validated using known clinical samples? Or is it a research use only kit that's been validated using known clinical samples? It's a mixture of blues and yellows. So there is some direction from other regulatory bodies, or it's developing tests the same, and that's very similar across different sites. I presume, and maybe Simon will touch on this as well, is that when you go through the if you do take in a kit from, say it's an FDA cleared kit, I presume there is then validation steps that you then take on yourselves, because obviously in a slightly different country there's an odds there. Question 3 is, if you were implementing and developing a new test, which one of the following samples do you currently use for validating your molecular diagnostic test? Is it, so in the red card, internal historical clinical samples? Is it external historical clinical samples, so this from your own hospital or from a collaborator or another hospital? Is it yellow, which is plasmids or oligonucleotides, or green, which is commercial reference materials? So we've got some green and red, we've got green and blues, greens and yellows, so it's sort of a mixture, a real mixture so a lot of people are using commercial reference materials, which is reassuring for Horizon, because that's what we do. It's also, it is important, and I think this is when we talk to people, is validation. With validation, you need to be able to understand what your test is doing, which is where the fully characterized reference materials is useful, because you can understand the limit detection, specificity, sensitivity. But it's also critical to use clinical samples, and so it's not you use this A or B. It's using clinical samples in conjunction with precisely validated reference materials. Question 4, how often do you validate your molecular diagnostic tests once you've set it up? So is it, the red card is you only validate at the time of setting up the test, the blue is you validate it with a sample every time you run the test, yellow is once a week, or green is once a month. Wow, okay, so everybody, so it's a mixture, well, yeah, it's a whole mixture. Some, one person that sets up just at the start, and then others that are running the control to validate the assay every single time. And that may be also reflective of a lot of you are doing next generation sequencing, which, due to the expense of that next generation sequencing and also the bioinformatic pipeline, having that control to validate the whole process is obviously critical. If you're doing qPCR or Sanger sequencing, you've probably been running that for a certain amount of years, and there may be more comfort in the robustness of those assays. Question 5 is, which feature of a molecular diagnostic test is most important to you? In the red card is the speed of doing the test, the blue is the cost of the test, the yellow is the availability of the test, or the green is the simplicity of the test. It's quite a tough question because everybody wants to put all the cards up, but this is we do have David who develops these kits. Does love this answer because yes, they are all important.
It's a very complicated answer.
So then I've sneaked in a question 6 of 5, and this is what's that? These on the phone. Why do you do what you do? So on the red card is driving the commercial success of your hospital or company. The blue is ensure liquidity of your lab or hospital and balance the budget correctly. On the yellow card is driving clinical research forward to improve patient survival rates, or the green is providing the patient with the best chance of survival and treatment. So we've got, you guys are just cheating now that you kind of like they're obviously all important. They are obviously all important because you do need to certainly keep the hospital running effectively, and likewise on the patient side of things, both driving clinical research and pushing forward the patient treatment is critical as well. So thank you very much for participating in that. I think that was probably, we've already got, I don't know if you've got any photos of that, but I think that was the most, you have definitely participated in more than everybody else, so I have to congratulate you as being the best audience so far on the trip. That then brings me back to the question that I was asking just before we did that, into what makes a good diagnostic assay. And what I've done is split diagnostic assay into 2 parts, because that's how it works. And you've got on one side you've got the pre-analytical part of the diagnostic assay, and on the other side you've got the analytical part of the assay. In the pre-analytical, what you have is you're taking a biopsy, you need to process the FFPE, DNA extraction, you may need to store it or transport it depending on if the sample collection is in a different place to the analytical part, and then you do DNA quantification. And the pre-analytical part of the process will control your DNA quantity and quality. On the analytical side of things, and which will lead to the treatment decision, is you have your DNA sample, there'll be some sample preparation. If you're doing Sanger sequencing, it'll be a PCR before sequencing it. If it's next generation sequencing, there'll be a library preparation step, and it'll be a bit more complicated. Then there's the analysis, and then that will lead to an actionable decision. And this will then control the quality of the diagnostic result. And together, you need very good pre-analytical steps as well as very good analytical steps. Now, what I've got next is some data from the College of American Pathologists. They ran an EGFR scheme last year to 226 labs, and at the same time they asked a number of questions. And one question they asked was, how do you quantify the DNA in your diagnostic test? And I think it was as expected that the majority of the labs do use nanodrop. It's a very easy method, very quick method to measure DNA. one thing to bear in mind, and I'm sure you all know that as well, is anything that's below 10 nanograms per microliter concentration, the nanodrop will overestimate the amount of DNA. So there is always the risk, certainly if you're working, going into next-generation sequencing, that you could underload the amount of DNA that you're putting into your assay.
John, can I also add to that actually the nanodrop only doesn't measure the total DNA, it doesn't differentiate between double strand and single strand. That's true. So that's why it overestimates. That's true.
And it's if there was another, using say a fluorometric method is a better way of quantifying the DNA. However, I also understand that it takes a lot longer and it's more expensive. So until there's sort of something to compete with nanodrop, I can't imagine that the lab cells would be swapping those. It's good to understand. I think the important thing is just good to understand the limitations of the different methods or different platforms you are using. For me, what was more astonishing about the results of this survey is you've got 16% of people that actually do not quantify their DNA before they put it onto their platform. So they extract the DNA from their biopsy using whichever kit, and then they, depending on what their standard operating procedure says, is they may take 5 microliters of that or 10 microliters of that and then put it onto their assay. And this just opens up the risk that you could underload the assay to not have enough DNA, or you might not even have any DNA, or even overload the assay. So you've actually put too much DNA. If you underload, you're likely to get false negative results. If you overload, there's a chance of false positive results. So it was a very curious result, and I think there's something that CAP need to address there within for the labs that are not quantifying DNA. They also asked a very similar question to the one I asked you, and I think results are very similar in that we've got the majority of labs are running Sanger sequencing or a qPCR method. They did distinguish between qPCR and commercial kits. I imagine a lot of these commercial kits are qPCR based, so actually a lot of, very similar here, there's a lot of labs using qPCR. The NGS, certainly for clinical diagnostics, is relatively small now still, but we are expected to obviously ramp up. Validating NGS pipelines is a very difficult thing because of the breadth of information you get and breadth of mutation coverage you need to test, and I'm sure that may be some challenges that you also face in setting up NGS here. Another bit of data that I wanted to pull out, and Simon or Dr. Page may touch on later, is exon 18 and 20 testing. And so as you often see the resistance mutations, the EGFR resistance mutations in there. And out of the 226 labs, 1 in 4 of those labs do not test for these mutations. So you may find that it's got an EGFR mutation that is treatable, but then they're not testing to see if there's a resistance mutation as well. So those patients could be potentially will be put on completely the wrong treatment that is could give them side effects, and it definitely won't be extending their life. And so it's the worst possible scenario for those patients. On the contrary, the labs that are testing are actually very good at it. So there's a 98% accuracy of those tests when they do analyse these exons. And so there's an educational component again required for the labs not testing, that actually they should test and get into this level of accuracy. So to summarise a little bit on the different types of platforms, and this is I imagine this is well known by you already, this is the limit of detection. So typically home brew methods or LDTs that are qPCR based, they are normally about 1% sensitivity. Likewise with qPCR commercial kits, normally at 1%. It is important that if you are using commercial kits, that you do also look at their claims, because I always assume that qPCR commercial kits are all at 1%, but actually the FDA cleared QIAGEN TheraScreen kit for EGFR. There's a couple of mutations in there that have got sensitivity of around 15%, and so it's important to understand exactly what your test is doing. Next generation sequencing is typically around 5% at the moment, likewise with Sequinone, the Roche Cobas pyrosequencing. Sanger sequencing, it does range from what mutations you're looking at, and it'll probably be it can be from 15% to 25%. the take-home message from this slide, and what I don't want this slide to be doing is kind of saying, right, well, you're sitting there, and actually what we want to be doing is we all want to be in this range. We all want to be doing like LDTs or qPCR. Sanger sequencing is a very robust method, and this slide is more about helping you to understand what does your platform do, or what does your pipeline do, what is the limit detection of your pipeline. Because Sanger sequencing is a very good method for detecting SNPs or deletions. And by knowing the limit detection, so if you know that your limit detection is 20%, if you were to get a biopsy sample that they'd cut into the normal tissue, so it was 90% normal tissue, 10% tumour tissue, you would know straight away that, okay, well, there's a likelihood that that 10% tumour tissue will be heterozygous, so the allele burden of that sample will be 5%. So you already know that, okay, if it is this sample, if I was going to extract the DNA from that complete sample, I'm not going to be able to detect that mutation with Sanger sequencing. So what I need to do is then do a micro dissection of that sample to then enrich for the tumour proportion. And once you've done that, you then know, okay, well, I've got 90%, 100% of the tumour sample, so I can still use Sanger sequencing, and I will, if the mutation is there, I can definitely detect it. Some of the labs might want to just go the other way, where it's like, actually, I regularly get 90% normal sample, so I just want a different method so I don't have to keep doing micro dissections, and that's then the choice again of the lab. A little bit on the a bit more on EGFR, and this is Simon will talk more about this, so I'll just touch on this. This was an EQA scheme sent out to, I think, in the region of 330 labs worldwide, and what you're looking at here is the percentage of incorrect results across 18 or so samples. The good parts are when there's no errors, so there's no errors for these samples, and then the bad points are where you've got, so for EGFR G71S, you've got 36% of labs actually got that sample, which could be false negatives, could be false positives, could be calling the wrong mutation. There's also wild type samples where they managed to find mutations that weren't there, and that's 1 in 5, that's 20%. And so if these are patient samples, they will be getting the wrong treatment. They'll get that report will be going to the clinician, and that clinician will be deciding, triaging their treatment, and it wouldn't be the best for them. Like I said, Simon will touch more on that. Now, what I do like to do is to try and bring the challenges that we've got in molecular diagnostics into a more real life experience, just to look at it from a different angle. And so, if you were sat on an aeroplane travelling to Heathrow, and a pilot came over the tannoy, it was like, "Good afternoon, ladies and gentlemen. We've got some very good flying conditions. It might be bumpy on the way, but we expect to arrive in Heathrow at 5 p.m. Next day. By the way, our altimeter has an error rate of 10%. There'll be some people on the plane who will be like, okay, don't really understand what that means, but the captain knows what he's doing, so we'll just trust the captain. There'll be other people, and probably a lot of you will be like, okay, what does 10% mean of error rate? Now, if you're flying into Heathrow, which is 25 metres above sea level, that just means that you're going to be coming in either 2.5 metres too high or 2.5 metres too low, and so you're going to be coming in quite hard, and it will be a bumpy landing. It's not too bad, but it won't be comfortable. If you're going to Guangxi, you've got that 690 metres above above sea level, so that you're going to be coming in plus or minus 69 metres. That's going to be a crash landing. That'll be touch and go whether or not you actually would be walking off that plane. Now, if you're going to base camp in Tenzing, that's 2,860 metres above sea level, which would mean that you're 286 metres out, and you're probably actually landing somewhere on Everest, unfortunately. And for me, it's like, I mean, it's a cute little example, but if you won't accept 10% error on an altimeter in an aeroplane, why would anybody accept 10% error for a molecular diagnostic test, which is going to be controlling how a patient is going to get their treatment, and it will be driving the chances of that patient actually living longer than 3 weeks, 4 weeks, 3 months, and whatever.